All right, scientific method. Go ahead and title your page scientific method. First things first, let's go through uh, what the scientific method is. It's a series of steps that allow us to test a hypothesis and solve a problem. Those steps include observation, hypothesis, experiment, data collection, conclusion, and then we do everything all over again when we retest. So starting with observation or problem, an observation is something that a scientist or really anyone would gather through their senses. What you see, what you hear, what you feel. So you notice something in your natural world, and you'd ask a question. So an observation is stated as a question. What you ask has to be testable, so it can't be based on an opinion. So it can't be are bluebirds prettier than cardinals? Prettier is an opinion. But could you test, do bluebirds um, sing on a higher octave than cardinals? You could test that. So here's your example. How does wavelength or color of light affect the rate of growth for pea plants? So that's an example. And if we look down at the bottom, and hopefully you guys remember this from chemistry or IPC, when we look at white light, just light from the sun, if we put it through a prism, like a raindrop, then we get a rainbow, right? And so that separates into all these different colors. And when we separate into all these different colors, it's based upon uh, the waves of those photons. So the wavelength of the photons and how they travel. So the wavelengths are higher for red light and lower for violet blue light. Okay. And that's what colors we see. Now, you have to understand that we see colors that are almost rejected from surfaces. So, in other words, reflected back at us. So, if you're looking at someone's blue shirt, that shirt is absorbing every other color but blue. And blue is the color it's rejecting, it's reflecting back out into the atmosphere, and you're seeing that color. If you're seeing red, that is what's reflected. Everything else is absorbed. Okay. So here's an experiment setup. And what you should notice is that, first of all, we're using the same types of plant. So spinach here, spinach here. We're going to have the same light source for both. So those are our constants. The only thing that we're going to change is that over here, we're going to use red light, and over here, we're going to use green light. Okay. Now, hypothesis. A hypothesis is a testable explanation for a question or a problem. So essentially, it's the answer to the question. It can't be an opinion. But if your question is not based off an opinion, then chances are your hypothesis won't be an opinion either. When you write a hypothesis, you don't want to use pronouns. So you don't want to use he, she, they, I, it. You pretty much want to stay away from it. Um, so starting off with, I think that blah, 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 take out the I think. Take out the I think. Just continue on with the blah, 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 whatever it is that you're going to say. Okay. But just take out the I think um, or we think, take all that out. Okay. So take yourself out of the experiment. So the best hypotheses are written in an if-then statement. And when you write a hypothesis in an if-then statement, then it's very easy to identify the independent and dependent variable. Because the way that the independent dependent variable go in the statement is if the independent variable, then the dependent variable. So you can figure that out real easy. The hypothesis is going to predict an outcome. It's going to tell us what could happen. Now, can a hypothesis be wrong? Absolutely. So nobody cares if your hypothesis is wrong because it's a guess. All right, so here's your example. If grown under red light, then spinach plants will have a greater rate of growth as opposed to those grown under green light. Okay, so you notice in the very beginning here, we have the light color. That's our independent variable, light color. And then we have rate of growth after the then, that's our dependent variable. Okay. If-then statements can be tricky to write. So if you're having a hard time writing one, you could just write a sentence. Spinach plants will have a greater rate of growth when grown under red light rather than green light. And that's perfectly fine too. Next is going to be the experiment. This is what you're going to do when you're trying to test your hypothesis. So you're going to number your steps. And again, you're not going to use pronouns. You're not going to say, number one, I placed the plant under the light. No. Okay. Number one, place 3 plants under the green light, 3 plants under the red light. Okay. And then you're just going to continue. All right. So when you're experimenting, the best thing to do is to only change one factor, only have one independent variable. Okay. So everything else stays constant. So the only thing that we're changing in this experiment is light color. We're not changing the amount of water. So each plant gets the same amount of water. We're not changing the plants. They're all the same plant. Okay. So we want to make sure that everything else is constant. That way we know that there's not other factors that influence plant growth. We want to know that if it's light. So variable is a factor in an experiment that's being tested. Again, you only want one variable. So typically you'd have a control group. That's a group that does not get the experimental, whatever the experimental, whatever the experiment is, like whatever is going on. So in this situation, we have red light, we have green light. Maybe our control group could be no light. Okay, so the idea behind a control group is that we're trying to compare, trying to make sure that light is even a factor at all. Okay, so the control could be that a spinach plant is not placed under any light. Or maybe the spinach plant is placed under white light. So again, the independent variable is what we're changing. Make sure you highlight that. The factor that is changed is known as the independent variable. Very important that you are able to identify that. My highlighting is not that great here. And the factor that we measure is a dependent variable. Okay, so in this situation, it is the wavelength of light that is the independent variable at the red or green light. And dependent variable is going to be the rate of change or the height of the plant. A valid experiment has to be repeatable. You have to be able to do it over and over and over again, which of course we could do this one over and over again. Okay, so typically you'll have two groups. The control group, which gets nothing, right? So maybe it's the plant that doesn't get light. And the experimental groups, those are the plants that are on the green and uh, red light. So notice here, under the green light on this side, we didn't have much growth there, right? So it's just kind of sad. But over here in the red light, it's like, whoo, lots of spinach, yum, salad. So a lot went on there. So in your mind, you're thinking, well, that doesn't make a whole lot of sense. Why is it that plants are green? Shouldn't they like green light? But remember what I said about light and color. Color is what you see when an object reflects back at you. So essentially, plants don't like green because they are green. They're actually reflecting the color green back out, spitting it back out because they don't want it. They want anything but green. Everything else is absorbed. So over here, red light was absorbed, green light was spit back out. And so since plants require light, these guys over here on this side did not get the same amount of energy as these guys over here. So data is information gathered from an experiment. Okay, so it can be quantitative, like our experiment, which means numbers, or it could be qualitative, which means pictures. Okay, so here's our quantitative data. So under red light, spinach plant 1, 2, and 3, we have about 0.centimeters per day for growth rate. And under green light, spinach 1, 2, and 3, we have about 0.1. So like I said, the red light is going to win because plants spit out green light. And then we would take that data from a chart and put it into a graph. Okay, and this is going to be a double bar graph. And what you're seeing here is right here, those are the plants grown under red light. We have a little legend right here, very important if you're going to have multiple bars. And these guys right here, these bars, plants grown under green light. When you're done, you can write a conclusion. Okay, so this answers your hypothesis. The best part of a conclusion, or the parts of the conclusion that are very important, starting with the claim. The claim is just a hypothesis rewritten so that it's correct. Okay, so rewritten so it's correct. Or you could just simply rewrite your hypothesis and in evidence accept or reject it. So if it's right, the hypothesis is accepted. If it's wrong, the hypothesis is rejected. Okay, then you're going to use the data here to prove your claim. Okay, so this does require you to use numbers. Okay, numbers. If you're using quantitative data, qualitative, you can use description, but quantitative data, you have to have numbers in the sentences. Then you need to give a scientific explanation for why what happened. And that's your reasoning. Claim, evidence, reasoning. All right, here's your example. So here's the claim. Spinach plants will have a greater rate of growth when grown under red light rather than green light. Okay, so there's the claim, and that's true. This hypothesis is accepted because it's true. Because the plants grown under the red light experience a growth rate of 0.6 centimeters per day. Notice how there is a number there, okay? Whereas plants grown under green light experience a rate of only 0.1 centimeters. Again, numbers, that's your evidence. And then here's your scientific reasoning. The pigment chlorophyll and chloroplast of plant cells absorbs the red wavelength and transmits or reflects back the green wavelength. Therefore, red light provides the cell more usable energy in the form of absorbed photons, than green light. Okay, and that's the truth. That's the reason why. And so here's a little graph so that you kind of understand how chlorophyll works. And you guys remember chlorophyll and chloroplast in plant cells. So chlorophyll is going to absorb all of this. Okay, so your purple and blues and all of this, your reds. But notice down here, your greens, it don't like green. And I don't like yellow. Not too much. And not so much on the orange either. Okay, so it's going to be red, blue, purple. That's what plants want. Those are the colors they want. These are the colors they're going to reflect back out. Okay. All right. So when you're all done, you're going to do it again. You're going to retest in order to verify results. And you do it over and over again. And once you get the same results over and over again, you can get a theory. Okay, so same results arrived at through repeated testing over time. So observation, hypothesis, experiment, you've got an experimental group, control group. You get results. Either the results are accepted or the hypothesis is accepted. Okay, so supported and accepted. Then you repeat and do this all over again, over and over and over again, right? Or hypothesis is not accepted. You revise it, come up with a new prediction, experiment, do it over and over again. Once you get to this place right here, and you're going over and over and over again, and then you finally do it enough times, get the same results every time, you can get a scientific theory. So just to kind of go back through it, identify a problem, state an observation about the problem, form a hypothesis, design an experiment to test the hypothesis, collect the data, form a conclusion, and do it all over again. This is the scientific method.